Welcome. Now let's proceed to the genus Eimeria. So Eimeria is a genus of Apicomplexa parasites that invade the intestinal tract of its host, causing enteric diseases or coccidiosis. Coccidiosis is an acute invasion and destruction of the intestinal mucosa. The clinical signs include fever, diarrhea, inappetence, weight loss, and emaciation. So we have here the different species of Eimeria for poultry. And we have here for the cattle, goat, and pigs. For the morphology, Eimeria oocysts are oval-shaped or elliptical and are typically 10 to 40 microns in size. For the structure, the oocyst is well protected from potential dangers in the surroundings by a tough cell wall. Thus, wall-forming structures are contained in the cell, as well as polar caps, micropiles, and other bodies. For the life cycle, most of Eimeria species are monoxenous, meaning their life cycle is completed within a single host. And when we say monoxenous, the parasites lives on only one kind of host. At the same time, they are stenocenous or host-specific. When we say host-specific or stenocenous, they only affect a certain kind of host or one kind of host. Now, we have here the diagram showing the three sequential life stages of the parasite. First one, we have the asexual sporogony or the forming of sporocysts containing infective sporozoites. During this process, oocysts undergo sporogony in the external environment, in which a process requiring oxygen, humidity, and favorable temperature. Next is the endogenous multiplication by asexual merogony or schizogony. Once inside the lumen of the gastrointestinal tract, the sporozoites exist from the ingested oocyst. The sporozoites then enter the host cells and give rise to the merons. Merozoites form inside the merons are released to reinvade the host cell, then repeat the process of merogony. And last, we have the sexual gamogony. After completing a series of merogony, merozoites penetrate the host cells and start the process of gamogony. Eventually, zygote will be formed by the fusion of gametes, and zygotes transform into an oocyst, which is shed through the feces of the host. Then the cycle repeats. For the pathogenicity, the development of oocysts to infective sporulated oocysts or sporulation does not occur above 40 degrees Celsius or below 20 degrees Celsius. So we have here the temperature range for the development. The sporulation occurs rapidly in less than 16 hours at temperatures between 30 degrees Celsius and 37 degrees Celsius. Sporulation occurs outside the host, requires several days and oxygen. Sporulated oocysts are resistant to adverse environmental conditions and can survive as long as 1 year in moist, protective environments if they are not exposed to freezing or extremely high temperatures. So we have here also the diagram for an example of the coccidiosis, specifically bovine coccidiosis. So the infection results from ingestion of infective oocysts. The oocysts enter the environment in the form of feces of an infected host, but the oocysts of Eimeria are unsporulated and therefore not infective when passed in the feces. Developmental stages reside in either cells lining the intestinal vilus or the enterocytes, or cells within the lamina propria of the villus. Maturation and emergence of asexual and sexual stages from infected cells causes cell lysis. This damage can be especially severe when caused by species that developed within cells in the lamina propria. Zoids are also found in the extraintestinal tissues such as mesenteric lymph nodes, liver, or spleen of the definitive host. These resting or latent stages are not thought to cause any clinical diseases. So for the diagnosis, infections are usually diagnosed by the coprological examination of host feces for coccidial oocyst or the concentrated using various sedimentation flotation techniques. Fecal examination should be performed using centrifugal flotation and an adequate amount of feces. For the treatment and prevention, sulfadimethoxin is the only drug that is label approved for the treatment of enteritis associated with coccidiosis. We also have here the ponazuril, which among the newer drugs, ponazuril appears to be effective according to published research and user testimonials. For the additional preventive measures, the daily removal of feces, disinfectants containing high concentration of ammonia can destroy oocysts. Steam and pressure washing may help to dislodge feces from surfaces. And lastly, we have the treatment of all in-contact animals may also be beneficial in controlling coccidiosis. Thank you.